So thank you for having me at this conference. My name is Carrie Holt and I'm a research scientist with Fisheries and Oceans Canada. And I'm going to speak about genetic risks of hatchery enhancement for Pacific salmon. And first, I'd like to acknowledge that I'm speaking from the traditional territory of the Tsuut'ina First Nation, which is part of the Coast Salish people in Nanaimo on Vancouver Island. I'd also like to acknowledge my co-authors on the work that I'm going to present today. These are Ruth Withler, Mike Bradford, and Dave Willis. And we worked together to develop a research document in 2018 on genetically based targets for enhanced contributions to Canadian Pacific Chinook salmon populations that I'll be speaking about today. So for an outline, first I'll give a short context on the objectives for hatchery enhancement, and then I'll talk about guidance about what the wild salmon policy, what guidance the wild salmon policy gives on hatchery management. And then I'll talk about some trade-offs in the management of hatcheries and the genetic risks of hatcheries and how managers can minimize these genetic risks within integrated hatcheries. And then I'll talk about one metric of genetic risks, the proportionate natural influence, or PNI. And I'll talk about how PNI can be used to designate populations influenced by hatcheries and to assess the biological status. So to start off with, I'm going to give a slightly broader context to this work. This work on genetic risks really pertains to hatcheries for a variety of different objectives. So SEP has 5 objectives for harvest, rebuilding, assessment, conservation, stewardship, and education. So for harvest, this is enhancement for fisheries that are reliant on enhanced production and would disappear or become severely constrained in the absence of enhancement. And so this includes harvest opportunities for First Nations, recreational, commercial fisheries. In terms of rebuilding objectives, this includes enhancement of stocks that are below apparent carrying capacity and includes rebuilding of depleted populations and mitigating for habitat loss. For conservation, this includes enhancements of stocks that are highly at risk of extirpation in order to prevent the extirpation and to preserve the genetic integrity or diversity of the population. In terms of assessment, this is fish production for the purposes of achieving assessment objectives and other objectives like around the Pacific Salmon Treaty for marking fish. And this objective is often used in combination with other objectives. And then finally, the stewardship and education objective, and this is fish produced for stewardship or educational purposes at levels that are considered to have low risks of impacts on natural production or other populations. So the genetic risks tend to be much lower for hatcheries in the stewardship and education, but it's still worth monitoring and keeping in mind. So in terms of the wild salmon policy and the guidance that this policy provides, the goal of the policy is to restore and maintain healthy and diverse salmon populations and their habitats for the benefit and enjoyment of people of Canada in perpetuity. So this policy defines wild salmon as an entire life cycle in the natural environment and were produced by parents that also spawned in the natural environment and lived there. So this schematic on the right shows that. So with the red circle, we can see an example of a fish that was produced in the hatchery. And for its first generation, it will spawn in the hatchery, go out into the ocean, and it can return to the natural environment. And we call these hatchery origin spawners. So they may be spawning in the natural environment, but they're from the hatchery. Their offspring, the second generation or blue circle, we call natural origin spawners. So they were born in the natural environment and they will return to the natural environment, but their parents would be hatchery origin spawners. And then this third circle, this third generation, we called wild spawners. So these are fish that spawn in the natural environment from parents that were also natural origin spawners. So that's our definition of a wild spawner under the wild salmon policy. The wild salmon policy also defines a conservation unit as a group of wild salmon sufficiently isolated from other groups that, if lost, are unlikely to recolonize naturally within an acceptable timeframe, such as a human lifetime. So there's enormous diversity of Pacific salmon. This map on the right shows the conservation units for one species, chum salmon, in British Columbia. There are over 400 conservation units among the 5 salmon species, Pacific salmon. I mentioned the goal of the wild salmon policy. There are 3 objectives underneath that goal. To safeguard the genetic diversity of wild Pacific salmon is one objective. Maintain habitat and ecosystem integrity is the second. And the third is to manage fisheries for sustainable benefits. So I have some text here from the policy that says that enhancement may be used to provide harvest opportunities and fishery benefits as part of an integrated strategic plan. So that gets at that third objective on the right, but also that enhancement, particularly hatcheries, poses some acknowledged risks to wild salmon. So this shows that there's kind of a balance here of enhancement that can impact and support fisheries, but there's also some risks to wild salmon. So as in fisheries planning, where there are trade-offs with abundances and trade-offs with abundance and trends are made to allow for socioeconomic benefits to be met, there's also a trade-off between the genetic risk from enhancement and the ability to meet social and economic objectives. So higher genetic risks have potential impacts on the conservation of wild salmon populations or conservation units. So we see this balance between genetic risks and the social economic benefits. So I'm just going to kind of provide some examples of these trade-offs for a variety of hatcheries. This is an example of a harvest objective hatchery, West Coast Vancouver Island Chinook. And this plot shows a time series of the terminal abundances over time from 1980s to 2016. So this shows an example of some of these trade-offs where the terminal abundance of Chinook is dominated by hatchery origin fish. So the black bars show the contribution of the hatchery fish versus other natural origin. And the terminal catch by First Nations recreational and commercial sectors are in the tens of thousands of fish. So, about half, in 2015, it was 56,000. So about half of that is terminal catch. So supporting this level of harvest has required very high inputs of hatchery fish. So this has resulted in trade-offs with the wild population values, but the potential long-term consequences need to be better defined and understood in terms of those genetic risks. This is another example showing some trade-offs for conservation and rebuilding objective hatchery for Saginaw Lake sockeye salmon escapement. This is a time series of the natural number of spawners. And you can note on the y-axis here that this is a logarithmic scale, so each line it goes up tenfold. So we can see that abundances are around 5,000 fish between the 40s to the late 80s, and then drop down to near zero. And when hatchery releases began, they contributed a large portion to the production. So the risk of enhancing at very high levels, after around 2000, was deemed to outweigh the risk of not enhancing, which would be the extirpation of the CU. So in this case, and others like it, the persistence of a population that resembles the wild population has implicitly been considered to be worth the trade-off of very high levels of hatchery salmon in the population. Then for hatcheries with an assessment objective, the production of hatchery juveniles for release with coded wire tags used to support domestic and international fishery planning and management. And coded wire tag release numbers required for a desired precision. So you need to release enough of these in order to get a precise enough abundance estimates, can result in high proportion of hatchery fish in the spawning return. Usually it's quite a bit lower than this, but occasionally it can get quite high. And for stewardship, hatcheries with stewardship objectives, These generally are small-scale production to support local conservation values and educational activities, such as salmon is in the classroom. And these two types of hatchery productions are generally understood to accept low risk tolerance, but better understanding is, better understanding is required to ensure that this is the case. So now I'm going to talk a little bit about what are these genetic risks. There's the potential for genetic risks to wild fish has been recognized since the inception of large-scale hatchery production in Canada and US. And these can be categorized as kind of demographic effects and fitness effects. This type of genetic alteration. In terms of demographics, demographic effects can occur when non-representative or too few fish are used as hatchery broodstock. Too few fish in hatchery broodstock. When there's non-representative hatchery broodstock, so that much of the diversity in the original natural spawning population is lost. And this is especially occurs when the broodstock produce high levels of return and there are compounding effects of reselecting fish from the hatchery families as broodstock. And there's been kind of an early research focus on managing that unintended loss of diversity in terms of choosing broodstock that's representative, choosing representative numbers and sex ratio for that brood. The second type of genetic impacts are related to fitness. And this is when these occur because there's an altered environment in which hatchery fish are spawned and reared. It reduces the survival of fecundity. So the fish that are born in the hatchery environment have reduced survival when they spawn in the natural environment. There's an increasing number of multi-generational studies of returning adults produced by hatchery origin and natural origin fish, and there are new tools available for measuring the fitness or reproductive success in both the hatchery and the natural environment. So we can use genetic tools such as parentage-based tagging to measure the reproductive success of those hatchery origin fish that spawn in the natural environment. And from those studies, there's a consistent demonstration of a reduced contribution of hatchery origin fish spawning in the wild, but there's some variability among species, populations, and brood years. So when we look at this reduced fitness of hatchery origin fish, first I'll define what I mean by fitness. This is the relative number of offspring left by an individual after one generation, and it can be influenced by the environment. We can use parentage analysis to measure the adult returns from the hatchery origin, or HO, and natural origin, or NO, fish spawning in both environments, both the hatchery and the natural environment. Then we can assign the returning adult the progeny to their parents in the previous generation to calculate what the parental lifetime contribution is from each of those fish from both the hatchery and natural environment. So we use a metric called the relative reproductive success, or RRS, of the hatchery origin fish in the natural environment to measure this. And this is a metric that at a value of 1 means that the hatchery origin fish reproduce as successfully as the natural origin fish in the natural environment. So that's when they have equal reproductive success. If a value of 0 or less than 1 means that the hatchery origin fish they produce less successfully, and at 0, the hatchery origin fish do not reproduce in the natural environment. If value is greater than 1, then the hatchery origin fish reproduce more successfully than the natural origin fish in the natural environment. So I'm going to give an study that measured this relative reproductive success of the hatchery origin fish in the natural environment. This is a study from Washington State where they assessed 10,000 genotypes of spawners from 2004 to 2006, and they found that this relative reproductive success was around 0.5 or slightly less. It was a bit lower for males and females, and overall was 0.45. This means that hatchery origin fish were much less successful than natural origin fish in the natural environment. Interestingly, they also found that hatchery origin homing was less accurate, and the hatchery fish homing to the natural spawning watershed was only 70% of that of the natural spawn fish. So now I'm going to talk a little bit about why we have this reduced fitness of these hatchery origin fish. And I'll talk about three things: the environment, genetic selection, and epigenetic selection. So in terms of the environment, the environmental influences, that these tend to be related to phenotypic plasticity. That is where the environment affects the phenotype. So although these are generally reversible, they can drive some heritable genetic and epigenetic alterations. For example, hatchery origin fish tend to be younger, tend to return at younger ages, and to return to lower areas of a watershed than wild fish, which explains some of their lower fitness. Genetic selection is another reason why they have reduced fitness. And this can result from altered or relaxed selection in the hatchery environment. And it can come from altered mating patterns. And this generally has long-lasting impacts. And the third is epigenetic mechanisms. And these are those that take place in the packaging or the structure of the DNA in the cells, and they cannot be detected from changes in the DNA sequence, but they can have profound effects on the phenotype of an individual by altering the expression level of the genes. So these epigenetic factors can be considered a hybrid class of mechanisms. They are often induced by the environment, but they do show some heritability, that is, some persistence over generations. Therefore, they're not as immediately reversible as the environmental influences are. So in BC, we have integrated hatcheries. Integrated hatcheries contain a single population that spawns in two environments, the hatchery environment and the natural environment. It's incorrect to view the hatchery origin and the natural origin fish in the river system as having different or genetically distinct groups because they spawn together in both environments. That is, both the hatchery origin and the natural origin fish spawn in both the hatchery and natural environments, creating constant gene flow between the two environments. This means that the broodstock is taken in part from the natural environment, and hatchery origin fish spawn in the wild environment. One benefit of having an integrated population is that gene flow between the spawning environment mitigates or slows down domestication, but does not prevent it. This differs from segregated hatcheries in the US, where broodstock is taken only from hatchery origin fish. There are two adaptive extremes in these integrated hatcheries, the natural environment and the hatchery, and an equilibrium exists, which balances the gene flow and selective pressures. These two environments are very different, and they exert different and opposing selective pressures on the fish as they experience that environment. This adaptive equilibrium exists, but perhaps not in a constant state, at which point the fish in the population will have the greatest fitness. The adaptive equilibrium will be closest to the hatchery optimum or the natural optimum based on which environment produces the most spawners in each generation. So this simple heuristic demonstrates this effect. On the first row, the adaptive extreme, the hatchery environment and the natural environment. And then the second row, this is the equilibrium population, which is in between the two. However, productivity matters. When the hatchery origin fish are more productive, or the hatchery produces a large amount of fish, then the adaptive equilibrium shifts towards the hatchery origin phenotype. As the entire population becomes more adapted to the hatchery environment, it becomes less well adapted to the natural environment and less reproductively successful in nature, reducing its contribution from that environment. The equilibrium state may have low fitness, but high hatchery production keeps abundances high, masking the unsustainability of the natural origin spawners, especially when unmarked, when hatchery fish are unmarked in the natural environment. It may reach a point that even while the natural environment is fully seeded with spawners, so hatchery origin spawners, it does not constitute a population that would be self-sustaining in the absence of the hatchery. Broodstock management also matters. Hatchery adaptation can be limited by maximizing the natural origin spawners in the broodstock. So in this schematic, we see that the hatchery origin brood are taken, the broodstock is taken apart from natural origin fish. So here we can keep the genetic contribution from the natural origin fish in the population dominant and minimizing the contribution of the hatchery origin fish. So there are long-term genetic consequences for both the local integrated population and for the regional or the larger level. In terms of the local integrated population, there can be lower genetic diversity and reduced fitness of the population in the natural environment. And this can apply to both the conservation and the harvest programs. At the regional or CU level, straying from enhanced population can reduce the reproductive success in the surrounding wild populations. Regional genetic diversity can be reduced and low natural origin abundances can be obscured. Genetic homogenization can result with reduced natural productivity. And under high levels of enhancement, wild salmon can be absent. So what are some ways that we can minimize these genetic risks? For hatcheries for harvest assessment and stewardship, we can ensure that hatchery production is scaled to the natural production of the system. We can use natural origin fish as brood to avoid domestication. And we can limit spawning of hatchery origin fish on the natural spawning grounds and control straying of fish into other populations outside of the integrated hatchery system. For hatcheries for rebuilding and conservation, we can produce as few hatchery fish as needed to supplement spawning in the natural environment as the population recovers. We can reduce hatchery production as the population rebuilds. And we can ensure that habitat and ecosystems are sufficiently healthy to support an increase in salmon production. Another thing that we can do to mitigate these risks is through a better understanding of an assessment of the genetic risks. So one way we can do that is through this proportionate natural influence metric, or PNI, where PNI is an indicator of the effects of hatchery production on the natural spawners based on gene flow between the two environments. So this is a metric that varies between 0 and 1, where at 0, the adaptations are dominated by the hatchery environment, and near 1, the adaptations are dominated by the natural environment. PNI is calculated based on PNOB, which is the proportion of natural origin broodstock. So within your broodstock, what proportion of those fish are natural origin fish? And PHOS, or the proportion of hatchery origin spawners. So among your spawners on the spawning grounds, what proportion are hatchery origin fish? So this PNI is not a measure of fitness or productivity changes per se, but may be an indicator of the potential genetic risk. So can we use PNI to classify populations into wild versus non-wild? So we've been starting to consider PNI in our biological assessments under the wild salmon policy in DFO, and Coast Seawick has also considered these in their assessments of Pacific salmon designatable units. As you recall from the wild salmon policy, that a wild salmon are those with a second generation, they are fish that are second generation natural origin spawners. So a natural origin fish paired with a natural origin and mating produces a wild salmon. However, hatchery origin fish paired with a natural origin fish or a hatchery origin fish paired with a hatchery origin fish, they do not produce wild salmon. So our classification needs to be stricter than simply a 50% of natural versus hatchery origin spawners, because we need to have wild salmon in our population. As a 50% would give only a small proportion of the wild salmon, assuming random mating. So develop some provisional guidance for integrated hatchery populations in assessments under the wild salmon policy by developing these 5 categories based in part on the PNI values. And these range from the top, these are populations that are adapted to the natural environment, and the bottom, these are populations that are adapted to the hatchery environment. So these top 2 have no integrated hatcheries. This is a wild population or a wild population that's influenced by strays. And then this third one, a category C, we call integrated wild, with PNI values then greater than 7.2. And this is where hatchery, within these populations, hatchery production is managed so that wild fish are greater than 50%. So these are second generational natural origin spawners, wild. Here the goal is high natural productivity. This fourth category we call integrated transition with a PNI value of greater than 0.5. Here, natural origin fish predominate, but wild fish are in the minority. So over 50% are natural origin, but less than 50 are wild. And for these, there's only moderate prospects for viability. And the bottom category are the integrated hatchery populations with PNI values of less than 0.5. And most fish are hatchery origin in these populations with current viability of low. These population categories can either define current status or they can be a long-term goal. For example, a population that's being enhanced for conservation reasons may have low PNI values, but have a long-term goal of high natural productivity. So have a kind of high PNI goal. So we've provided some provisional guidance for integrated hatchery populations for conservation programs. In some conservation situations, PNI standards may be relaxed in early phases to allow a sufficient hatchery production to bring the population out of critical low levels. And recovery to wild or integrated wild designation is not possible if habitat and other threats are not mitigated. So hatchery production cannot be used alone. Hatchery must be part of a broader recovery program to address the causes of decline. And PNI is a useful measure of the role of hatchery in recovery. The Salmon Enhancement program is now considering PNI in its programming. PNI designations are being considered for incorporation into production planning processes, where it's possible to assign biological designations, the A through E, those 5 categories I described before, to each production line to set PNI objectives. The biological designation would be considered jointly with the production objectives in a structured decision-making process. So this would enable management of the population in a way that could best achieve the socioeconomic objectives, as well as meeting biological goals around the fitness and diversity. In practice, the population biological objectives will serve as both a goal and an assessment of the current status. We can also apply these PNI metrics to biological assessments within DFO. The recommendation so far would be for biological status assessments of wild salmon to include the wild and integrated wild populations, and provisionally the integrated transition and wild stray influence populations. However, populations designated as integrated hatchery, that's that 5th category, would not be considered in the biological status of wild salmon. Since integrated hatcheries contain only a single population that is either adapted to the hatchery environment or natural environment, recall that it's one population, not two, the entire population is either removed or included in the assessments. And there's the recommendation to include PNI in the biological assessments of populations. The future steps include identifying PNI objectives for populations, identifying management actions to meet those PNI targets, and to develop monitoring programs for adaptive hatchery management. And I just wanted to highlight this excerpt from the Minister's Mandate level from January that discusses developing a Pacific salmon strategy. And so these metrics and consideration of genetic risk could form part of that salmon strategy. I want to thank you for your time. Sepka, which is thank you in Hokaminam, the language of the Sunyamuk people. And I'll leave with this slide showing some of the references used in this presentation. Thank you very much.